Most fungi live unseen as microscopic spores and filaments in the environment around us, where they play an essential and complex role in the ecosystem. Although we may not know it, we are in constant contact with fungi. For example, we breathe in airborne fungal spores every day. Some fungi live in humans as members of the microbial communities that live in our guts and on our skin. These microbes support our health in many ways, including helping to regulate our immune systems. However, a handful of fungal species have the potential to cause deadly human disease. For most people, exposure to disease-causing fungi is uneventful because a healthy immune response efficiently kills invading fungi. Fungi generally cause infections only when our skin or membranes are physically breached, for example, via insertion of intravenous catheters in hospital, or when our immune system becomes compromised. In the absence of a robust immune response, fungi can grow in the body, spread through the bloodstream, and invade and survive inside otherwise healthy human tissue. In this way, fungal infections can become established, causing significant tissue damage and resulting in severe diseases of the lungs, the brain, or the kidneys, for example. To invade human tissues, fungi which infect us from the environment must be able to recognize the new surroundings in which they find themselves and swiftly adapt to them. And those fungi which are normally living on or in the human body need to adapt their behavior to become invasive and cause an infection. Adaptations of this kind are triggered by switching on and off specific fungal genes, which instruct infecting fungi to produce the proteins, such as toxins and enzymes, that are needed to invade tissues. Collectively, the adaptation mechanisms that enable fungi to cause disease are called virulence mechanisms. As they grow, many disease-causing fungi are able to change their cell size and shape. Some small rounded yeasts can form giant cells, and yeasts and inhaled fungal spores can form long filaments. These changes in cell shape help the fungal cells to penetrate blood vessels and spread within tissues, causing masses called lesions that result from fungal growth and cause an influx of immune cells to the infection site. This inflammatory response is elicited to clear the fungus, but it does come at the cost of damaging host tissue, which, if unchecked, can ultimately lead to organ failure. For this reason, understanding the processes and outcomes of fungal infection and inflammation in humans is a critical goal for medical mycologists. Fungal diseases are difficult to diagnose, and better tests are required to detect fungal infections. And there are only a few effective treatments for fungal diseases, and these few therapies are at risk of becoming ineffective because of increasing antifungal drug resistance. Therefore, new types of antifungal drug must be developed. Researchers at the MRC Centre for Medical Mycology at the University of Exeter are studying fungal biology and the immune system to better understand how fungi cause disease, and are using this information to design better diagnostic tools to develop new antifungal treatments, including immune-based treatments. Visit the Our Research pages of our website to learn more about the work we are doing to help us learn more about what tips the balance in the battle for survival between fungal pathogen and human host.